Hi guys, I just wanted to walk you back through, we are in general biology, so you click general biology class, go down to modules, so click modules and scroll down, and we are in this week's material, so they're labeled by date now, March 30th through April 5th, cellular biology notes and resources. I want you to scroll down inside that and go to the study guide for exam 3, cellular biology. Then when you get that opened, go to file and make a copy, and that will open up a copy for yourself so that you can go through and put in your own answers and have a really great study guide for the future exam questions. So the parts we've already covered are the eukaryotic cell with the different types of organelles and their function, so how you would recognize what they are. So for example, this is a Golgi apparatus, so number 6 on here would be Golgi apparatus. And its function is to package up proteins, modify them, and put them out in little vesicles for the cell to carry out elsewhere. So there are a lot of generic bubbles, so 4, 12, and 10. There's another 10 down here. Those, they're just generic bubbles, so you can call them vesicles, lysosomes, or peroxisomes. We won't deduct if there's just a generic bubble inside a cell. You really can't know without a key what was intended for that one, because there's not a distinct shape. But number 13 is the centriole. Its function is to produce spindle fibers. You kind of see like a little bit of number 11. It's really pale, but 11 is just labeling the cytosol, or the jelly goo that suspends all of the organelles inside the cell. And number 9, so you can see a closed version of number 9 and the open version of the mitochondria. So number 9, whether it's cut in half in a cross section or whether it's closed, those are both mitochondria, which are the powerhouse of the cell. So the function of the mitochondria is to produce ATP. So sugar is broken down and then converted to a usable energy source to do work inside the cell, and that's adenosine triphosphate, or ATP. This number 8 is the smooth endoplasmic reticulum. The rough ER is right here, or rough endoplasmic reticulum, and the difference between the smooth ER and the rough ER is that the rough ER, or number 5 here, is studded with ribosomes. So ribosomes are actually proteins that are specifically folded to be able to build other proteins. So ribosomes aren't just little dots, they're actually those complex tertiary structures of protein that can put amino acids together fairly well. So they form those peptide bonds and make other proteins. Those are housed right outside the nucleus. So number 2 here is the nucleus, and inside the nucleus is the nucleolus. So the nucleolus actually contains the information to build ribosomes. So that's why there's like a concentrated little area inside the nucleus. But everywhere else in the nucleus would be DNA, our normal DNA, to write all of our other proteins. So the nucleolus is a concentrated area that does kind of look like a yolk inside the nucleus. And I think I labeled all of that, so I just want you to be able to go through that and be able to fill out this chart. Then hopefully you can, with the next assignment this week, compare prokaryotic to eukaryotic cells. So prokaryotic is bacteria and archaea. So archaea are ancient bacteria. So they live in extreme environments, but prokaryotic just means that it's before nuclei. So pro is before, and karya, so eukaryotic means true kernel. So true kernel means that it has a nucleus, and this is before there was a true nucleus. Okay, so fill out that chart in the next assignment. And I wanted to kind of skip over meiosis, mitosis to go to healthy cell growth and healthy cell division. So we can type in there some characteristics for the cell. So healthy cell division is going to be controlled. I can type. So, and then the contradiction to that, or the opposite of that, is cancer cell growth, which is uncontrolled cell division. So when we are back to healthy cell growth and division, it's going to be controlled. It's going to produce specialized cells. So that just means that a liver cell will continue to be a liver cell. So we'll put in a line here. And this, when it's a cancer cell growth, it will divide. So a liver cell could divide, and liver cancer is not really a thing, but sometimes I guess it does, good cancers. But say it's skin cancer or something like that, or breast cancer, the tissue will start dividing and reproducing at the cellular level, and it won't specialize. So unspecialized cells. You can tell that it came from, like, since I used liver over there, but it will just kind of be not itself. So they can still have the same genetic material, but it will just be a cancer cell after it's just kind of a blob. So that's kind of one of the easier parts of cancer for us to detect. So melanoma, you'd see a change in the texture of your moles, or the coloration would change. For breast cancer, you're supposed to feel for lumps, and the reason that you can feel for those lumps for a testicular cancer or breast cancer is because it's going to feel more like a pebble or some kind of foreign object under the skin, because they're not specializing. Those cells are not healthy. They're not as fleshy and full as a normal cell, so they kind of do form a layer of protection by them not specializing, because we can detect them better. That's definitely not healthy cell division. These healthy cells can also decide when to die. So there's programmed cell death. Different cells in the body are normally programmed to die. So in embryonic development, we have webbing in between our fingers and toes, and our body just tells it that, oh, okay, so we don't want webs in between our fingers and toes. We will just naturally kill off those cells. And for cancer, they are immortal. They don't ever tell themselves to die. They just keep living on in that uncontrolled, unprogrammed way. Contact inhibition here. Let's say that you do get a cut and you're healing back together again. Well, when one skin cell bumps into another skin cell, it's going to say, oh, we don't need any more cells here, we've healed up. Whereas the cancer cell growth will just keep growing. And reproducing. So again, that's something that wouldn't happen in a cancer cell. It would just keep going. That's part of it being uncontrolled, right? And then when we have healthy cell growth and division, we have repair that's helpful. And when we have cancer, it's a sickness in and of itself. So being uncontrolled, it can lead, obviously, cancer is a pathology, right? So, but sickness can lead to metastasis, which is a word for when something like a pancreatic cell decides that it wants to divide uncontrollably. Then once that pancreatic cell releases into the bloodstream, and then it can go produce cancer in other tissues throughout the body, that is metastasis. So once like a breast cancer or something like that moves to the lymphatic system, and the lymphatic system then can go travel to other parts of the body, that is metastasis. So it can spread. And they can still tell where the cancer started through genetics. So they can do testing for where that cancer came from. But once it has metastasized and spread to other tissues, it's much, much harder to stop and heal. So healthy cell growth and division is, just normal part of life for us growing up. We get bigger, obviously we need new cells, or when we are using and breaking down our cells all the time. So just think of how much skin cells you must go through, as there's wind, rain, loofah pads, it's like there's all kinds of things wearing away at our skin. So we need to produce healthy new skin cells all the time. So that's normal, healthy skin cell development and division. But then when we have cancerous cell growth and division, it's not controlled, it's not healing at all, and it is a normal part of life. So some cells do break down and just start reproducing, and our body has lots of coping mechanisms to digest those cells, break them down, and stop them from becoming the true pathological cancer. But then when those systems and buffers wear out, then we do get cancer in the body. So those are the differences between normal cell growth and cancerous cell growth and division. So thank you very much, and we will get on to meiosis and mitosis in our next video.